Welcome to the deep dive. Today, our material, it forces us to look beyond just the clinical science. We're staring directly at maybe the most fundamental question of human existence: fate versus choice.
That's right, because the scientific journey we're tracing today, it's really deeply rooted in human suffering. Our sources, they don't start in some pristine lab. They start with a family struggle trying to understand unpredictable illness. Detailing the lives of two brothers, Rajesh and Jagu.
And the story of Rajesh is particularly haunting, isn't it? In 1946, he apparently just underwent the sudden personality shift. His volatility, his emotional range became extreme. Good news would trigger these uncontained outbursts of joy, but bad news, it plunged him into this desolate blackness. The sources describe it almost like a sine curve, of his psyche, tightening in frequency, gaining in amplitude, and then ultimately tipping into rage and grandiosity. It sounds terrifying.
It does. And then his brother, Jagu, he faced something different but equally devastating: a slow, agonizing cognitive decay. His whole world just dissolved into paranoia.
He thought the local banana vendor was recording him.
Yeah, things like that. And he'd obsessively recite these completely made-up train schedules, like Shimla to Howrah by Kalka Mail. Just a desperate kind of failed attempt to impose some order on a mind that was clearly losing its grip.
Wow. When you hear those details, that whole question of fate, of destiny, it becomes intensely personal, doesn't it?
Absolutely.
So what is that fundamental, irreducible unit that underpins such complex and often just cruel human outcomes? Our mission today really is to trace this concept of the gene, that basic biological instruction.
Right. From ancient philosophical guesswork.
All the way through the double helix discovery.
And right up to the modern tech that lets us not just read these instructions, but actually start to, write them.
It's a huge journey spanning, over 2 millennia?
Easily. And for most of that history, before we had microscopes or biochemistry, people were just grasping at straws, weren't they? Trying to figure out how parents pass traits to kids.
And it's kind of astonishing how early the social engineering aspects started. Way back, 380 BC, Plato, apparently influenced by Pythagoras, suggested children were just arithmetic derivatives of their parents, like a math problem.
Right. And from that purely mathematical theory, Plato derived this, profoundly authoritarian idea that state guardians could somehow decipher and enforce the law of births.
To what end?
The goal was guaranteeing the production of the fittest children. It led directly to what the sources call numerological eugenics, based on numbers.
That's horrifyingly prescriptive. But thankfully, Aristotle, he was more grounded, wasn't he? His analytical approach kind of pushed back on this speculative stuff.
He did. Aristotle systematically dismantled that popular idea of the time, the traveling library notion of inheritance.
The idea that semen gathered instructions from every single body part.
Exactly. He pointed out logical flaws, like men pass on traits they don't have yet, like beards, or even intangible things like a specific way of walking. Those non-corporeal traits, they just couldn't sort of materialize into genetic material to be collected.
Makes sense. But even with Aristotle's logic, this other idea, the homunculus, it persisted for centuries, didn't it?
Oh, yeah. It was visually very seductive.
This fantasy, even after the microscope was invented, that a tiny, fully formed human being was just perfectly coiled up inside every sperm.
It's such an appealing idea because it simplifies everything. The process isn't really creation or generation, it's just propagation.
Like unfolding something that's already there.
Exactly. As the Dutch scientist Swammerdam wrote, in nature there is no generation, but only propagation. The new life was just a tiny version that simply had to get bigger.
Okay, let's jump forward. Mid-19th century. Darwin and Wallace discover natural selection, the engine of evolution. But this is where Darwin hits that huge wall. He admitted the mechanism of heredity was a very wide blank.
Yeah, the big problem was the dominant idea at the time: blending inheritance.
Like mixing paint.
Exactly. If traits just blended, like mixing buckets of colored paint, then any new useful variant, a helpful mutation, it would just get diluted out by the normal population over generations.
Into colorless oblivion, as the source puts it.
Right. Natural selection couldn't work if the advantageous trait just kept getting watered down. Darwin's theory needed a way to fix that altered trait to keep it distinct.
And Darwin, needing a fix for his huge theory, he came up with, a genuinely flawed solution, pangenesis.
Yes. This was a pretty crude hypothesis, genesis from everything. It proposed these minute particles called gemmules were supposedly thrown adrift by every single cell in the body, collecting and carrying hereditary instructions back to the sex cells.
So wait, Every part of my body, my eye, my liver, my left elbow, is like packaging up tiny instruction manuals and sending them off to my reproductive system, hoping they get passed on.
That was the idea, like little internal biological messengers. Darwin himself admitted it was a rash and crude hypothesis, but apparently it temporarily brought him considerable relief.
Huh. It's sort of comforting in a way that even Darwin got the core mechanics of inheritance fundamentally wrong at first.
It really is, which brings us neatly into part 2, where the mechanical pieces finally start to snap into place. And the key breakthrough, it came from a place Darwin couldn't really see. The microscopic level. The single organism.
Gregor Mendel.
The genius of Gregor Mendel. Working away quietly in a monastery garden. He wasn't asking about evolution over millennia. He asked a much smaller but crucial question. How does a single organism transmit information to its offspring just over a single generation?
And his pea experiments just shattered that blending theory, didn't they?
Completely.
Yeah.
When he crossed tall plants with dwarf plants, he didn't get medium plants. He got only tall ones. The shortness didn't become intermediate. It just vanished in that generation.
Which disproved the central objection to Darwin's work right there.
Exactly. But then when he bred those hybrid tall plants together,
shortness came back.
It reappeared, perfectly intact in the third generation. Mendel realized traits don't blend. They're discrete units. He called the overriding trait dominant and the hidden one recessive. The hybrid plant was a composite, carrying both sets of instructions, ready to pass them on.
So these discrete units, people eventually saw them.
They did. By the early 1900s, scientists like Boveri and Sutton, they located these units, these genes as they'd come to be called, on the filamentous chromosomes inside the cell nucleus, visualized like beads on a string.
Okay, so they had a physical location strung along chromosomes, but they still didn't know what the beads themselves were made of, right?
That was the chemical question. And interestingly, it fascinated the quantum physicist Erwin Schrödinger. In 1944, he theorized that the gene must be this molecule of contradictions.
How so?
It had to be chemically regular enough to be copied perfectly of trillions of times in cell division, but also capable of extraordinary irregularity to account for all the diversity of life on Earth. Regularity and irregularity in one molecule.
And the answer arrived almost right after that.
Pretty much. Oswald Avery's transformation experiments, also in 1944, proved definitively that DNA, not protein, was the material carrier of genetic information. The identity was finally chemical. Deoxyribonucleic acid.
DNA. And once they knew how the structure was the next big step.
Which led to the iconic 1953 breakthrough. Watson, Crick, Wilkins, and Franklin solving the double helix structure.
It's such a famous image.
And for good reason. The complementary base pairing A, always with T, G always with C, it didn't just look elegant. It immediately suggested the exact copying mechanism needed for heredity, how one strand could serve as a template for the other.
And that copying mechanism led straight to the functional map, what Francis Crick called the central dogma.
Exactly. This idea that biological information flows systematically. DNA holds the master instructions.
To build RNA.
Which acts as the messenger.
To build proteins.
And proteins are the workhorses, the machines that execute the instructions, enabling structure and function. And this flow, it's universal, from bacteria to elephants, as the source says.
But this incredible knowledge of the gene, it was co-opted almost immediately, wasn't it? Which brings us into part 3. And it's a tragic reminder that this fundamental knowledge, it doesn't exist in an ethical vacuum.
No, it certainly doesn't. This chapter really belongs to Francis Galton, Darwin's cousin. He coined the term eugenics.
Which means?
Essentially, the self-declared betterment of the human race through artificial selection. He genuinely believed what nature does blindly, slowly, and ruthlessly, man may do providently, quickly, and kindly.
That idea that humans can step in and actively improve the species.
It took root incredibly quickly, particularly in 1920s America, and it was fueled by this intense race anxiety over mass foreign immigration. And it wasn't just confined to labs, it became a kind of grassroots public movement.
You see that so vividly in things like the Better Babies contests held at agricultural fairs.
Right. Children were literally placed on pedestals and judged like prize cattle. The standardized score sheets actually instructed judges to score 50% for heredity before they even looked at the child's physical appearance.
Wow, 50% just based on who their parents were.
Yeah. And in this whole political and scientific push, it culminated in the 1927 Supreme Court case, Buck v. Bell.
Which did what?
It affirmed the state's right to sterilize individuals deemed unfit. The court's validation of the premise was just chillingly succinct. 3 generations of imbeciles is enough.
It's truly horrifying to think the highest court played a role in justifying that kind of violation of human rights. And the tragedy just amplified from there, didn't it?
It did. The Nazis explicitly borrowed the outlines of their 1933 sterilization law directly from the American eugenics program. But they intensified it, mandating that direct force may be used for compliance.
And the propaganda.
The propaganda was designed to dehumanize. Films like Das Erbe or Erbkrank show these stark contrasts. Images of individuals they labeled defectives. Like, there's a description of a woman with shortened limbs walking on all fours set against celebrations of the perfect Aryan body, like in Riefenstahl's Olympia.
And then came the slide from sterilization to outright murder.
Yes. Once Hitler authorized euthanasia for genetically defective children, the euphemism Lebenswertes, Lebenswert worthy of living, was used to justify mass extermination.
Facilitated by scientists themselves.
Tragically, yes. Scientific complicity was key. Joseph Mengele, who was actually a newly minted human geneticist, he became the angel of death at Auschwitz, performing these monstrous targeted experiments, particularly on twins, shouting "twins out" on the selection ramps. Genetics was twisted from a science of discovery into a tool for racial extermination.
Given that absolutely harrowing history, it feels almost miraculous that science regrouped and moved forward. Which brings us to part 4, the recombinant DNA revelation and the start of biological manufacturing.
Right. This revolution really kicked off in the 1970s. Scientists like Paul Berg, Stanley Cohen, Herb Boyer, they developed the tools to actually cut and paste DNA. Recombinant DNA.
And they use bacteria to do it.
They did. They used these circular mini chromosomes from bacteria called plasmids.
Plasmids. Let's pause there, maybe. Because they're crucial. They're basically like little biological flash drives, are they? Small loops of DNA that bacteria can pass around.
That's a great analogy, yeah. And scientists realized they could plug new instructions into these plasmids. And Stanley Cohen had this key insight. They could use natural selection in the lab to find the bacteria that had taken up the new DNA.
How did that work?
Well, say they cut an antibiotic resistance gene from one plasmid, one flash drive, and successfully pasted it into another plasmid. Then they expose a whole batch of bacteria to that antibiotic.
Only the ones that successfully incorporated the new plasmid with the resistance gene would survive.
Exactly. The antibiotic selects for the successful recombinants. They had effectively consigned evolution to the petri dish.
And Boyer immediately saw the business potential.
He did. Use these microbial factories to create human products.
Yeah.
There's the famous story. After about 3 hours and 3 beers with the venture capitalist Robert Swanson, they decided to found Genentech. That was 1976, aiming to manufacture life-saving drugs, like human insulin, using these gene splicing techniques.
A huge leap. And the next logical leap was trying to correct genetic disorders in humans, gene therapy.
Yes. The initial hope came in 1990 with Ashi da Silva. She was treated for ADA deficiency, a type of SCID, the bubble boy disease. The idea was to use a retrovirus as a vector, a delivery vehicle, to carry the corrected gene into her cells.
Yeah, and the results?
They were somewhat ambiguous, but encouraging for her and her family. But that cautious optimism just crashed violently in 1999, the death of 18-year-old Jesse Gelsener.
Right. He was in a trial for a different condition, OTC deficiency.
Yes. And he died just days after receiving a high dose of an adenovirus vector.
What went wrong?
The adenovirus vector, that biological delivery truck they used, it triggered a massive, uncontrollable, and ultimately lethal immune response in his body.
Oh, terrible.
And the investigation afterwards was just devastating. It revealed incompetence, serious side effects in previous patients that hadn't been properly reported, and crucially, the lead investigator had a massive personal financial stake in the company developing the therapy.
That conflict of interest, it's staggering. The gravity of that death, a young man entering a trial hoping to help others.
It sent the entire field of gene therapy into what the sources call a temporary free fall. A moratorium was called.
Yeah.
It was just a hard, painful lesson in the dangers of scientific hubris, conflicts of interest, and regulatory failure.
Absolutely. And that hard-earned knowledge, that caution, it leads us into part 5. Moving past just fixing disease to thinking about what genes actually dictate about human identity itself. And it's not simple cause and effect, is it?
Not at all. The functional map is much more complex. The sources give this equation: genotype plus environment plus triggers plus chance, phenotype.
So your genes plus your surroundings plus specific events plus just random luck equals you. Your observable traits.
Exactly. Genes provide a propensity, a tendency, not some kind of ironclad destiny.
One of the most fascinating examples of this is the biology of identity, looking at sex versus gender. The SRY gene is mentioned a single master switch on the Y chromosome.
Right. And a mutation in that one master switch can result in a person who is chromosomally male, XY, developing what the source calls a wholly female self, anatomically and psychologically. It's known as Swyer syndrome.
Wow. That really shows the unilateral power of a single gene to influence identity, but it's still interacting with development.
Precisely. It overrides the chromosomal blueprint in a profound way.
Okay, now let's talk about race. Genetics tells a very different story than history, or even how we often perceive it, doesn't it?
Absolutely. Genetics reveals that something like 85 to 90% of all human genetic diversity exists within so-called racial groups.
Within them, not between them.
Exactly. Only about 7% of genetic variation is between those traditionally defined groups. Our sources really emphasize that racial assignment, scientifically speaking, is pretty much a futile exercise. It's driven overwhelmingly by cultural history and social decisions, not deep genetic reality.
That's such an important point. And finally, what about behavior? Temperament.
This is where twin studies have been really revealing, especially identical twins reared apart. They showed these powerful correlations around 0.5 to 0.60 for personality traits, temperament, even attitudes.
And that's a correlation similar to physical traits like height or weight.
Yes, remarkably similar. But what they shared wasn't necessarily identical identities. It was more like the first derivative of identity, a shared propensity towards certain behaviors or ways of being.
Can you give an example?
Sure. The D4DR7 repeat variant gene is mentioned. It's linked to novelty-seeking or stimulation-seeking behavior. Okay. Now, that propensity could manifest as, say, focused, determined creativity in one twin. But in the other twin, raised in a different environment with different triggers and chances, it might manifest as risk-taking behavior or even addiction. Same underlying genetic propensity, very different outcomes.
That distinction between propensity and destiny is just crucial, isn't it? Especially because today we're gaining the power to step in and actually alter those propensities.
That's the precipice we're on. We're moving beyond negative eugenics, just selecting against disease, like with pre-implantation genetic diagnosis, PGD, and moving into the realm of potentially selecting for perceived improvement, positive eugenics.
And this is where CRISPR-Cas9 comes in, the gene editing technology.
Exactly. It's the ultimate toolkit, really. It allows geneticists to make incredibly precise directional changes in the DNA sequence. We're not just reading the code anymore. We can effectively write it, or at least edit it with high precision.
And the source mentions a scientist engineering 14 distinct alterations in human embryonic stem cells in just 6 months.
Yeah, that demonstrates the speed and power. That level of rapid, precise modification was just unthinkable even a decade or so ago.
And it's this capability, specifically the potential to add new information or make heritable changes to the human genome, that alarmed the scientific community, right?
It absolutely did. It led key figures like Jennifer Doudna and David Baltimore to call for a moratorium on germline editing.
Meaning changes that could be passed down to future generations.
Exactly. Changes to sperm, eggs, or embryos. They warned quite starkly that if we start justifying intentional alterations by trying to improve ourselves, then inevitably someone gets empowered to decide what constitutes an improvement.
And that leads us straight back to the hubris, the danger that fueled the entire eugenics movement in the first place.
Precisely. It's a profound ethical threshold. So we've traveled this immense journey from Plato's arithmetic children, through Darwin's misguided gemmules, past the discovery of DNA's perfect structure.
Through the terrible shadows of eugenics and the setbacks of early gene therapy.
We started this deep dive, thinking about the personal fates of Rajesh and Jagu, wondering about destiny, about what was written in their biology.
And now, as we gain the ability to intentionally alter the genome, the source says the circle of logic closes on itself. We started as observers of fate, maybe victims of it. Now we're gaining the tools to potentially become its architects.
Yeah. And the source material quotes a scientist who contemplates the human genome as potentially being manifest destiny. Think about the implication of that phrase.
It's huge. If we learn to eliminate all infirmities, all vulnerability, maybe we could mitigate chance. But in mitigating chance, do we also somehow mitigate choice? Do we engineer away the very things that make us human?
What biological, cultural, social precepts should guide us now that we, as an intelligent species, are learning not just to read the instructions for life, but potentially to write our own?
An infinitely deep question for the future. Thank you for joining us on this deep dive.